Welcome back to the new section. In the previous section, we have discussed in detail on the biochemical pathways with regards to the insulin and its effects. In this particular section, we are going to see the actual chemistry, that is the organic chemistry of carbohydrates and their reducing potential. You might be asking me why it is needed in medicine, but I feel they are really important as far as medicine is concerned because they are the ones that form the basis of our understanding of how this capillary blood glucose technique works and how your urinary blood glucose measurement works and how your plasma blood glucose measurements work and what are the pitfalls of the same. And you have to understand all these things to know whether the report that you're seeing is correct or not correct. And at the same time, it's also of paramount importance to understand why the diagnostic criteria for diabetes is there. So the diagnostic criteria of diabetes is basically two things. One is the excessive glucose and second is the excessive HbA1c, and how these things are going to correlate with your end organ damage and why this diagnostic criteria has been proposed. For that, you have to understand how this excessive glucose is going to have an impact on the body and how they are going to cause this microvascular and macrovascular complications. For that, you have to understand a term called glucotoxicity. We'll be discussing in detail about that in this video as well. And at the same time, we are going to kickstart our discussion with insulin receptor physiology, which we have not done in the previous video. So that is really important because insulin, upon its binding with the insulin receptor, is going to have multitude of effects, and certain effects can happen within seconds. So that is the reason understanding of the insulin receptor physiology is really, really important, where we are going to discuss a lot of pathways. Majority of them have already been discussed in the very first video of endocrinology. In this, we are going to just recap all those things in a much better way. And I'm also going to give a very important overview of another pathway called as AMP kinase pathway. That is important, especially for the mechanism of action of certain drugs like metformin, so which we'll be seeing in the same video as well. So let us start with our understanding on insulin receptor physiology first. To start with the insulin receptor, as we know it, it is a receptor tyrosine kinase. It's an RTK with this intrinsic tyrosine kinase activity. We know that upon its binding with its ligand, that is the insulin, it's going to result in activation of the tyrosine kinase activity and reactivity, which will result in autophosphorylation of the receptor itself. That's why it's called a receptor tyrosine kinase. Basically, this insulin receptor is a heterodimer where you're going to have two alpha domains and two beta domains. So it's a heterodimer basically. And once insulin binds in the insulin receptor, I told you there will be activation of tyrosine kinase activity, which is going to result in autophosphorylation of the receptor. Once autophosphorylation has happened, it is ready to phosphorylate its substrate. And we know what the substrate for this insulin receptor, that is called as IRS. The name is apt because it is insulin receptor substrate 1 and 2. So IRS 1 and 2 are basically the substrates for insulin receptor to be phosphorylated. Once IRS 1 and 2 are phosphorylated, they become active. And they're going to activate a lot of downstream pathways, out of which two pathways are important. One is the MAP kinase pathway, which is going to get activated. We know this is a pathway for cell proliferation. It's a mitogenic pathway, which we'll be discussing in some time. And IRS 1 and 2 can also activate another pathway called PI3K AKT pathway. So first they are going to activate this phosphatidylinositol 3 kinase, which is more metabolic in nature. Clear? So let us first discuss about the PI3K pathway in detail. So what are the downstream effects of PI3K? So once PI3K is activated, that is going to convert the phosphatidylinositol diphosphate to phosphatidylinositol triphosphate. Once PIP3 is formed, PIP3 is going to activate certain other downstream kinases. The first kinase that is activated is PDK1, which will be activated. This kinase is going to activate another terminal protein called as AKT. So this is the terminal protein that is activated, and that is the reason why this pathway is also referred to as a PI3K AKT pathway. And majority of the metabolic effects of insulin are actually mediated through the AKT proteins only. So what are the effects of AKT for that matters? So as I told you, AKT is going to be involved in a lot of metabolic effects with regards to insulin. So what are the metabolic effects is the question. So first effect, we know insulin is going to increase the glucose uptake by the cells. How they do that? Especially insulin responsive tissues like skeletal muscle, adipose tissue. These are going to take up glucose in response to insulin. So increased glucose uptake, that's the first effect. And how they're going to do that? By increasing the number of GLUT4 transporters on the surface. That's what we refer to as an increased GLUT4 translocation to the surface of the cells. How this is going to happen is the question. The molecular mechanisms is what I'm asking. So first of all, we have to understand there is a protein called AS160, which is actually trying to inhibit the GLUT4 receptor translocation. The vesicles that contain this GLUT4 receptors will not be allowed to move to the cell surface in the presence of this AS160 protein. Insulin, by activating PI3K-AKT pathway, is trying to inhibit this AS160. So whenever AKT is formed in excess under the influence of insulin, they are going to inhibit this AS160 protein, which is actually trying to increase the GLUT4 translocation to the surface, and that is how insulin is trying to increase the glucose uptake by the cells. And of course, this increased glucose that is taken up by the cells will be shunted towards glycolysis, which we have seen already. And of course, insulin is going to activate a variety of glycolytic enzymes as well, like fructose 1,6 bisphosphate, which you have seen already in the previous video itself. Now we are moving on to the second metabolic effect of insulin, that is going to increase in the glycogen synthesis. How they increase the glycogen synthesis? By stimulating an enzyme or stimulating the activity of an enzyme called as glycogen synthase. How they do that? First of all, let us understand there is one important kinase that is actually regulating like a glycogen synthase enzyme, that is called as GSK3, that is called glycogen synthase kinase 3. Glycogen synthase kinase 3 is going to actually inhibit glycogen synthase by actually phosphorylating them. What insulin is going to do is by activating the PI3K-AKT pathway, it is trying to inhibit the GSK3 enzyme. So AKT is a powerful inhibitor of glycogen synthase kinase 3, which in turn is going to become as a positive response in terms of glycogen synthase enzyme, where the activity of glycogen synthase enzyme will be actually increased. So this increased activity of glycogen synthase enzyme is the one that is responsible for increased glycogen synthesis with regards to insulin action, which is a very important action of insulin, which we have discussed in the previous video in detail itself. And the third effect, let us put as other effects to be precise. So we know one of the important effects of insulin is to increase the lipogenesis, that is the fat synthesis, free fatty acid synthesis in insulin responsive tissues like adipose and the liver. Let us see how that happens. So first they are going to stimulate the mTOR complex 1 through a variety of intermediate proteins are there, like tuberous sclerosis proteins are there, where they are going to stimulate the mTOR complex 1. So once mTOR complex 1 is stimulated, we know mTOR complex can increase the cell proliferation. mTOR complex can increase the protein synthesis, and we know insulin is anabolic. It makes the cell to synthesize more protein. And at the same time, it can reduce the autophagy, and this effect is mediated through a protein called ULK1, which we'll be discussing in the AMP kinase pathway. And at the same time, it's going to also stimulate the one important protein called SREBP1C. That's called a sterol responsive element binding protein 1C. So what this SREBP1C is going to do? So we know the central dogma of biology, where the DNA is going to result in mRNA, and the mRNA is going to get translated to certain proteins. So let us assume the proteins what we are discussing here are nothing but the enzymes of the fatty acid synthesis, like acetyl-CoA carboxylase and fatty acid synthase. And there should be an mRNA for that, and there should be certain genes for that. And these genes that are producing these enzymes of fatty acid synthesis are regulated by a very important area. This is a transcription regulation site that is called as SRE, that is called sterol responding element. So what is going to happen is the fact that this SREBP1C is going to be activated in the setting of high insulin thereby activating the PI3K AKT pathway. AKT pathway is going to stimulate the mTOR complex. mTOR complex in return is going to stimulate the SRBP1C. And this sterol responsive element binding protein 1C is going to bind with the sterol responsive element, thereby stimulating the transcription of these genes that are going to produce this acetyl-CoA carboxylase and fatty acid synthase, which in turn, of course, is going to increase the fatty acid synthesis, which means insulin is an anabolic hormone, which is going to increase the lipogenesis within the cells that are going to respond to insulin. We know that already, but we are just seeing the molecular mechanisms in this video. And apart from that, insulin can increase the activity of HMG-CoA reductase. Of course, we know HMG-CoA reductase is a very, very important, a crucial enzyme in the cholesterol synthesis pathway. So insulin can increase the cholesterol synthesis as well. And apart from that, there are some minor effects with regards to insulin. Like for example, they can increase the cell proliferation by inhibiting a pathway called FOXO1. So it's a very important pathway of research. Inhibiting FOXO1 will result in increased cell proliferation. And at the same time, they can also activate the endothelial nitric oxide synthase production, thereby producing vasodilation. So insulin also has vasodilatory effects. All right. So now we have discussed a series of physiological pathways that are found in relationship with the PI3K AKT pathway, which is one of the major pathways as far as insulin is concerned. Now we are moving on to the MAP kinase pathway. It's a minor pathway with regards to insulin, but still a very, very important one as well. So what is this MAP kinase pathway and what's going to happen here? So once the IRS1 and 2 is activated, let us start from the beginning. IRS1 and 2 is activated. That is going to stimulate and activate certain group of proteins, and they're going to phosphorylate them and activate it, called as GRB SOS proteins, which will be activated. And once GRB and SOS are activated, they are going to activate the very important mitogen called as RAS. We have discussed that already, which will be activated. And once RAS is activated, you are going to activate RAF group of proteins. You have ARAF, you have BRAF, the details of which we have discussed in the very first chapter in endocrinology itself. These are going to be activated. So as a cascade, now once RAF is activated, they are going to activate this MEC1 and 2. And once MEC is activated, they are also going to activate ERK1 and 2. Once ERK is activated, they are going to get translocated into the nucleus, where they will be activating the transcription of various other factors that are going to increase the cell proliferation, like CFOS, CJUN, CMYC, ETS, and so many other proteins. And these proteins are the ones that are collectively called as mitogens, which are going to be helpful in the cell proliferation and mitogenic activity of the cell. Of that, this RAF is also referred to as a MAP3 kinase, or otherwise I can write MAP-KKK, MAP3 kinase, and this MEC is what we refer to as MAP2 kinase. Either I can write MAP2K or MAP-KK. The third, this ERK is what we refer to as a family of kinases called as MAP kinase. Remember, these kinases have different other groups of proteins also. It's not like RAF is the only MAP3 kinase and MEC is the only MAP2 kinase and ERK is the only MAP kinase. We have other group of proteins within this family, but for our understanding, these three things will form our basis of understanding for advanced oncology later on. But anyways, so these three are very, very important proteins, and these are collectively called as MAP kinases. And that's why this pathway is also referred to as a RAS MAP kinase pathway. It is a mitogenic pathway, which is actually activated by the insulin mechanism. Fine. So after discussing about the insulin receptor physiology, we have to also discuss about one more important pathway that is called as AMP kinase pathway. But before that, I would also like to tell you that the insulin receptor and its autophosphorylation activity and its tyrosine kinase activity can be actually upregulated, or it can be activated by a very, very important protein called PPAR gamma. PPAR gamma stands for peroxisomal proliferator activator receptor gamma, and that is the reason why PPAR gamma activators like thiazolidinediones are going to increase the insulin sensitivity because they are going to increase the activity of the insulin receptor in general. Clear? And at the same time, there is one important entity that can actually inhibit the insulin receptor and its activity, that is diacylglycerol, that can actually downregulate the insulin receptor activity as well as they can actually downregulate the insulin signaling in the form of inhibiting the IRS1 and 2 phosphorylation. So that is the reason why obesity is a risk factor for developing insulin resistance. Whenever there is obesity, there will be increased free fatty acids. This increased free fatty acids will be converted to monacylglycerols and diacylglycerols. And diacylglycerol, as we know through research, is a powerful inhibitor of the insulin receptor autophosphorylation, subsequent phosphorylation of IRS1 and 2. So I have told you two mechanisms how PPAR, gamma and thiazolinions can stimulate the insulin receptor and improve the insulin sensitivity, and how fat, that obesity and diacylglycerol within the cell can actually downregulate the insulin receptor activity by inhibiting them, resulting in development of insulin resistance. Now the turn is to understand another pathway which I promised in the beginning, that is the AMP kinase pathway, which is going to behave like an antagonist in certain areas, not in all areas. In certain areas, it's going to behave like an antagonist to the PI3K-AKT pathway. We know that PI3K-AKT pathway basically is an anabolic pathway, which is happening in response to insulin binding with the insulin receptor, but whereas this AMP kinase pathway is going to act as a catabolic pathway predominantly. Because insulin is a hormone of well-fed state, PI3K-AKT pathway is anabolic, and at the same time this AMP kinase pathway is catabolic, which means it's going to happen in low energy states and starvation. Let us see what you mean by AMP kinase pathway. First, understand what is this AMPK. It's a protein. We know that it's a kinase which is going to be activated by none other than the AMP. That's why it's called the AMP kinase. More than the levels of AMP, it is the AMP-ATP ratio in the cell that's going to determine the activity of AMP kinase, where the increased levels of AMP-ATP ratio is going to result in activation of AMP kinase. So it doesn't mean that only the AMP level should increase. Even if the ATP levels are low, resulting in a relative excess of AMP can activate the AMP kinase. So what are the conditions that increase the AMP-ATP ratio within the cell? First is the exercise. Definitely they are going to increase the AMP-ATP ratio because exercise sucks out a lot of ATP and uses a lot of ATP, resulting in relatively more AMP compared to that of ATP, resulting in activation of AMP kinase. It's a physiological cue. Apart from that, you have certain drugs that can do this, like metformin. Metformin's majority of the actions are actually proposed to happen through the AMP kinase pathway only, even though there are some other hidden mechanisms that are with metformin which are yet to be delineated. But a lot of effects of metformin can be explained through activation of AMP kinase pathway. How it's going to do that? So metformin indirectly activates AMP kinase by inhibiting the complex 1 of electron transport chain. So thereby reducing the ATP production, because that results in mitochondrial uncoupling, resulting in reduced ATP production, resulting in relatively more amounts of AMP, increasing the AMP-ATP ratio, thereby activating the AMP kinase activity within the cell. So it indirectly is going to activate the AMP kinase activity. Apart from that, in low energy states like calorie restriction or starvational states, of course, it's going to reduce the ATP production, increase the AMP-ATP ratio, resulting in activation of AMP kinase. And there is a fourth trigger, that is a protein that can actually directly stimulate the AMP kinase, that is called LKB1, a very important question as far as exams are concerned. So once the AMP kinase pathway is activated, that is AMP kinase is activated, it's going to have a lot of effects and have a vigorous effects, in fact, on a lot of metabolisms happening within the cell. The first one is going to be the action on the glucose metabolism. In the glucose metabolism, the first effect is to increase the glucose uptake by causing more GLUT4 translocation. So thereby it's going to actually increase the glucose uptake by the cells and thereby shunting them towards glycolysis. Of course, this is important because this is a pathway that is going to be activated during exercise states, starvation states. So definitely you need more energy for the same, so that they try to increase the glucose uptake. And that's how metformin is also trying to improve the insulin sensitivity by causing more glucose uptake by the peripheral tissues. You would have studied that effect of metformin in pharmacology textbooks. That's how it happens. And apart from that, they can suppress the gluconeogenesis. And you can remember metformin, one of the important effects of metformin on blood glucose is to reduce the blood glucose by suppressing the hepatic gluconeogenesis. Very important word in pharmacology textbooks, which we'll be discussing again later on in the treatment of type 2 diabetes mellitus chapter. And third effect is they are going to reduce the glycogenesis by suppressing the glycogen synthase activity. So let us see the molecular mechanisms now. How GLUT4 translocation is increased. It's by stimulating a protein called TBC1D1. This protein can actually help in translocating more GLUT4 to the cell surface. AMP kinase is trying to activate this protein, thereby activating the GLUT4 translocation. And suppression of the gluconeogenesis happens through AMP kinase's ability to suppress certain gluconeogenic enzymes like PEPCK, that is phosphoenol pyruvate carboxykinase, and glucose-6 phosphatase, which is a very, very key enzyme with regards to the gluconeogenesis. At the same time, they can suppress the glycogenesis by reducing the activity of glycogen synthase enzyme. All right, clear? So these are the effects of AMP kinase on the glucose metabolism. Apart from that, it's also going to have effect on the lipid metabolism, where it's going to behave almost a total opposite to what PI3K-AKT pathway does, where it's going to be completely catabolic. First of all, it's going to inhibit the acetyl-CoA carboxylase enzyme, which is going to translate as reduced lipogenesis. They are not going to produce fatty acids in the setting of more AMP kinase. And second, they can also stimulate the hormone-sensitive lipase enzyme, where insulin is trying to suppress the hormone-sensitive lipase. AMP kinase is going to stimulate the hormone-sensitive lipase, which is going to translate as increased lipolysis. As I told you, the only regulated step in the beta oxidation of fatty acids is the lipolysis step. After that, the entire beta oxidation process is actually not regulated properly. So, of course, if you produce more lipolysis, you are going to have more free fatty acids, which will be entering the beta oxidation. And of course, all these things are needed because in the setting of low energy state and exercise, you should mobilize the energy stores to be utilized by the cells. So that's why all these things are happening, to increase the levels of ATP within the cell. And they're also going to suppress the activity of HMG-CoA reductase. And this is a very, very important, crucial enzyme as far as cholesterol synthesis is concerned. So thereby they are going to reduce the cholesterol synthesis as well. And this can be related to the metformin's weight losing potential, which is a very important one. That's what you have studied. Metformin can effectively produce weight loss, but it's not through the GLP-1 receptor or through some other action. It's by increasing the AMP kinase activity indirectly. And it can also have effect on the mTOR pathway, where they can actually suppress the mTOR pathway through activating a protein called TSC2, that's a tuberous sclerosis complex protein. But first, let us see what are the effects of mTOR proteins. First effect is to inhibit the apoptosis by down-regulating certain tumor suppressor proteins like P53 and P27. So this can result in reduced apoptosis. We know that mTOR is a cell proliferation stimulation pathway. So they can do that by suppressing the apoptosis. And at the same time, they can also increase the cell proliferation through a variety of pathways happening within the cell. That's why they are mitogenic pathways, and they can increase the protein synthesis as well within the cell. And of course, they are going to down-regulate the ULK1. So once they down-regulate the ULK1, which will be translated as reduced autophagy. ULK1 is a very important protein as far as autophagy is concerned. That is important for your pathology as well. As I told you before, mTOR pathway is going to be inhibited by the AMP kinase through a very important intermediate protein called TSC2, that is tuberous sclerosis complex protein number 2, where AMP kinase is going to stimulate the TSC2. TSC2 in return is going to inhibit the mTOR complex. We know the effects of mTOR complex. So inhibiting the mTOR complex means the opposite is what is going to happen here. So in this setting, there are going to be increased activity of the tumor suppressor proteins like P20, P27 and P53, which means there's going to be increased apoptosis, reduced cell proliferation, reduced protein synthesis, increased ULK1 and increased autophagy, clearly catabolic in nature. And at the same time, this AMP kinase pathway can also have a grasp on the mitochondrial metabolism, where they are trying to increase autophagy independently by activating ULK1 and 2. And they can also increase mitophagy, and finally they can increase the oxidative phosphorylation metabolism as well, resulting in more ATP production. And that's the basis of this entire pathway. This happens in the setting of low energy states, like low ATP states, which means they are trying to increase the ATP by mobilizing the energy stores, at the same time activating cellular metabolism that are destined to produce more ATP. Of course, AMP kinase is a catabolic pathway. We know that. And that's the major difference from the PI3K-AKT pathway. Even though you might be confused by the fact that AMP kinase pathway is trying to increase the glucose utilization, like what insulin does, but that's the only similarity. Apart from that, that glucose is actually utilized for anabolic functions within the cell in the presence of PI3K-AKT pathway, where that glucose that is utilized by the cell is actually used for catabolic reasons. Here, glycogen production will not happen. So comparison, PI3K-AKT pathway is going to happen in the setting of well-fed state, which means high insulin states, where the pathway is actually anabolic in nature, where AMP kinase pathway happens in the setting of low energy states or poorly fed states, where the entire pathway is supposed to be catabolic in nature. And final point is the fact that there are 3 subunits of AMP kinase. The alpha subunit is going to be the most important. Majority of the effects of AMP kinase are mediated through the alpha subunit only, and we have beta and gamma subunits as well, which play a minor role in this pathway. Now let us go to the Minardo plot, which is a very interesting plot, because insulin and its action within the cell is very complicated, with certain things happening within a few seconds and certain things happening within a few minutes, and it's really complicated. So that's why we are taking the help of the plot that was originally invented by Charles Joseph Minard, who's actually a French civil engineer who's known for picturizing a complicated information in a very simple diagram. His work is a genius, the work of a genius, which dated back to the 19th century, beginning of the 19th century, where in 1812, the Napoleon's troops were actually defeated by the Russian troops in the Battle of Tarantino. To be honest, it's exactly around October 16 or 18 in the 1812. So where he plotted that in a very, very beautiful way, in a single image. It's so good to see. So that's why history is really important, because when you know the history, it becomes really interesting and you become really happy to understand the present, what we are doing actually in the current era. So let us start with that first. So we know that Napoleon's troops started in France with a strength of 4,22,000. And this beach line is the one that tells you that the Napoleon's troops are moving towards the east, towards the Moscow, and the thickness of the line will tell you how much troops are left at every point. As they move towards the Moscow, you can see that the line is getting thinner and thinner, which means the amount of army that is left is decreasing and decreasing over a period of time. So where you can see once they have reached the Moscow, there are only 100,000, that is 1 lakh of the troops that is left, 1 lakh of the men that is left there. That is probably because the starvation on the way, and at the same time the extreme nature of the climate as well, where he has given the temperature fluctuations and the cold. That is also one of the reasons why the troops died on the way itself. And this black line will tell you the return path of the troops, and at the end, when they reached their original starting point, only 10,000 of their men left among the 422,000 that have started. And in a single plot, how beautifully he has given the forward march of the troops as well as the backward march of the troops back to the origin. And at the same time, he has given the amount of troops that left at each and every point, and he has depicted the time scale as well. He has told the temperature, and that is the major reason for the problems for the troops. And at the same time, he told the distance that is covered and the map as well in the single diagram. So that's why this has become a landmark plot, and this is one of the very, very important plots in statistics as well, because this is how you represent a complicated information in a simple pictorial manner. Similarly, insulin's action can also be demonstrated in a similar plot. That's why we are keeping a same name called as a Minardo plot, where once insulin binds with the insulin receptor, there is series of effects that are happening over a period of time, and it's too complicated to tell in a simple tabular column or something else. So that's why we are using this Minardo plot, where you can see within 15 to 30 seconds, there is priming of glycolysis, and there are so many reactions that are activated or inhibited in the process. So initial 15 to 30 seconds is for priming of glycolysis. And in the first 1 to 2 minutes, there is going to be inhibition of lipolysis through multiple metabolic pathways, and there is also going to be initiation of protein synthesis in 1 to 2 minutes. And in 2 to 5 minutes, there is going to be increased GLUT4 mediated transport of glucose into the cell, which means there is going to be increased glucose uptake at around 2 to 5 minutes. At around 5 to 10 minutes, your glycolysis will start happening. That will be accelerated within the cell. And at 10 to 20 minutes, your lactate will increase, because one of the important effects of increased insulin is increased production of lactate. And at the same time, at the end of 20 minutes, your glycogen production will increase and your lipogenesis also will increase. Clear? So, if you have time, you can go through the plot. It's such a beautiful plot, a complicated information, if just depicted in a very simple time scale. Let us move on to the fact that how glucose that is present in increased amounts in the blood can result in problems in the setting of diabetes mellitus. This is where your understanding of the glycolytic pathways becomes really, really important, because without understanding them, it's very difficult to understand anything in diabetes. That is the reason why I've explained in great depth and detail about the glycolytic pathways in the previous chapter itself. If you have not watched the previous chapter, please watch them and come back. So first of all, what is glucotoxicity? The effect is due to high glucose itself. That's the problem. High glucose can overwhelmingly activate the glycolytic pathways, producing more glycolytic intermediates and more energy production, number one. And at the same time, even after this increased activity of glycolytic pathways, this much amount of chronically elevated glucose cannot be absorbed by the glycolytic pathways, resulting in spilling over of this glucose to certain supplementary pathways and glycolytic intermediates to certain supplementary pathways. What are they? Let us see. First, increased glucose itself, which is not absorbed by the glycolysis, will be spilling over towards another very important supplementary pathway called as polyol pathway. This pathway will be active only in the setting of too much of glucose. It cannot be active in the setting of normal or low glucose. This can be active only in the setting of chronically elevated glucose, where glucose will be converted or reduced by an enzyme called aldose reductase, because glucose is an aldose, 6-carbon aldehyde. So where it will be reduced by this enzyme aldose reductase to an alcohol called sorbitol. And this sorbitol can be further processed to produce fructose, and in this step there is another important enzyme that is getting into act, that is sorbitol dehydrogenase enzyme. And both these steps itself comes with certain complications. At the same time, sorbitol per se is going to create certain issues. So what is the issue created by sorbitol? First of all, sorbitol, when it is produced in excess amounts, it's a very powerful osmotic agent which is going to draw water. Once sorbitol draws excessive water to the area where it is produced, and this is thought to be one of the important reasons for developing certain diabetes related complications like cataract and certain microvascular complications like diabetic neuropathy. Even though it has a role in diabetic retinopathy and nephropathy, but it's more important in the setting of diabetic neuropathy. So cataract, microvascular complications, sorbitol plays a very important role because of its osmotic properties. And apart from that, these two enzymes itself is going to create issue, because the first step of conversion to sorbitol is NADPH dependent process, which is actually going to deplete the NADPH stores in the body. Number one, NADPH is very important to prevent the cell from going for free radical mediated oxidative stress. But if there is low levels of NADPH in the cell will go for free radical stress and it might result in cellular damage. And that's what happening in G6PD deficiency, which you have seen already. Let me recap in some time. And the second problem that's going to result is because of sorbitol dehydrogenase step, because they are going to create excessive NADH from NAD. So what is NADH basically? NADH is an electron carrier. So already the cell is not able to cope with oxidative stress. Coupled with that, you are producing more amounts of NADH is obviously a problem, because these electron carriers, they are potential time bombs if you are not utilizing them properly. So they can produce this oxidative damage to the cell if these free electrons are released. So that's why production of NADH produces free radical stress and oxidative stress, and coupled with loss of NADPH in the cell will expose the cells to more free radical mediated stress. And we know who's going to produce this NADPH. that is your G6PD enzyme, glucose 6-phosphate dehydrogenase, where this glucose 6-phosphate will be forming 6-phosphogluconate, which is nothing but they are going to enter the HMP shunt to produce the ribose sugars for DNA synthesis. And of course, this G6PD is the one that is the rate limiting enzyme in the HMP shunt, and that's going to produce the NADPH, of course, and that will be utilized by the sorbitol and the polyol pathway. And you are going to have reduced NADPH, means you are going to have more oxidative stress. How is this possible? So we have discussed already in the hematology section. Let me recap it once again. So you have free radicals, which are nothing but oxygen with uneven electrons, and these uneven electrons means they are highly reactive and they always look for certain electrons to combine with, and they get neutralized. So once they combine with another electron, they change that compound to a free radical, and that's how the chain reaction keeps going on, and they cause damage to different number of molecules over a period of time. So these free radicals has to be neutralized, which means they have to be reduced. In order to make them reduce, you need to have a reduced compound which is ready to get itself oxidized, that is glutathione. So reduced glutathione comes into our protection, where it's going to neutralize these free radicals by reducing them. In the process, it itself is going to get oxidized. But this replenishment of the reduced glutathione is very, very important for reducing the oxidative stress in the future, and this replenishment of reduced glutathione will come in the presence of NADPH, which gets oxidized to produce NADP. But in this setting, NADPH donates this hydrogen to reduce this glutathione and maintaining the glutathione in reduced state, which means it can neutralize the free radicals in the future. Suppose if you do not have NADPH in the cell, of course you are going to result in loss of this reduced glutathione stores in the cell, which means your free radicals will go unchecked, and excessive free radicals will result in oxidative stress. They are nothing but electrons. They are going to result in oxidative stress, free electrons. And of course, NADH being energy-rich compound, which means they are electron carriers. If it's not processed properly by the TCA cycle, of course, they are going to result in more generation of free radicals, and that's why this polyol pathway is problem from multiple dimensions. Of course, the problem is not going to come only from the polyol pathway. There's going to be other problems from other glycolytic intermediates that are produced in excess. For example, there will be excess production of this fructose 6-phosphate as well, because the entire glycolysis is accelerated in setting of high glucose. Of course, we know this fructose 6-phosphate is a raw material for another pathway called as hexosamine pathway, where they will be forming something called glucosamine 6-phosphate. And for this, you need a very important enzyme called as GFPT, or you can call it as GFAT, where it stands for glucosamine fructose 6-phosphate aminotransferase. And this glucosamine 6-phosphate will be further processed and will be converted to a compound called UDP N-acetyl glucosamine. And this is going to serve as a raw material for a very, very important post-translational modification process called as glycosylation. So there is the thin line of difference between glycosylation and glycation. Both the processes, you're going to have addition of glucose to a protein. But if it happens enzymatically, we call it as glycosylation. If it happens non-enzymatically, we'll be calling it as glycation. So in this process, the addition of glucose to the protein happens enzymatically, and that's why we call this step as glycosylation step. And this is a normal post-translational modification that is happening. And remember, addition of glucose to a protein by a process called glycosylation can change the character of a protein to an extent. For example, it can either reduce the functional activity of a protein, or it can increase the functional activity of the protein, and that's why we have this post-translational modification in the first place. And depending on the needs of the cell, they can alter the functionality of a protein. In hyperglycemic states or in diabetes mellitus, whenever you are going to have excessive fructose 6-phosphate, means there will be excessive shunting of this fructose 6-phosphate towards the hexosamine pathway, and there will be excessive glycosylation and too much glycosylation that can alter the functionality of a lot of proteins in a much different way. For example, they can reduce the function of certain proteins like GLUT4, where too much glycosylation of GLUT4 transporters may result in reduced functionality, resulting in reduced glucose uptake, resulting in more hyperglycemia and more problems. And similarly, they can also result in increased functionality of certain proteins like TGF-beta. where, as I already told you, increased TGF-beta is very, very important for increase in the extracellular matrix production. Extracellular matrix is actually deposited by activity of TGF-beta only, which is a very important cytokine. And extracellular matrix deposition is really, really important for certain microvascular complications like diabetic nephropathy, which we'll be seeing subsequently. Apart from that, this glyceraldehyde 3-phosphate itself is a problem, because whenever glyceraldehyde 3-phosphate is produced in excess beyond the absorptive capacity of glycolysis, they can be shunted towards other parallel pathways. In this example, they can produce more methyl glyoxal, and this methyl glyoxal is also referred to as carbonyl glyoxal, one of the form of carbonyl glyoxal, because here the methyl group is the carbonyl group that we are talking about, and one of the forms of carbonyl glyoxal is methyl glyoxal. And excessive production of this carbonyl glyoxal compounds produces a stress called carbonyl stress within the cell, and that will result in the development of certain important products that are going to be discussed in some time, called as AG, called as advanced glycated end products. Production of AGs are normal as aging process starts, but it can be produced in more amounts in the setting of diabetes mellitus. And this AG is having a lot of effects locally as well as systemically to cause complications that are related to diabetes mellitus, which we'll be discussing subsequently. So increased glyceraldehyde 3-phosphate producing more methyl glyoxal, creating more carbonyl stress, and that is going to result in production of more advanced glycated end products, which itself is going to have a lot of issues. And at the same time, this excessive glyceraldehyde 3-phosphate can also be converted to produce more dihydroxyacetone phosphate, which will be shunted to produce more glycerol 3-phosphate. We all know that glycerol 3-phosphate is a very important raw material for the production of monocyl, diacyl and triacyl glycerols. Of course, the important compound that we need to talk about with respect to glucotoxicity is the excessive production of diacyl glycerol. You will be resulting in more triacyl glycerol and monocyl glycerol as well, but this diacyl glycerol in excess is what the key is. Diacyl glycerol can activate, because we have studied pharmacology, we have studied the endocrine basics in the beginning itself. We know that diacyl glycerol is a very powerful activator of a very important molecule called protein kinase C, especially the epsilon component, that is protein kinase C epsilon. Whenever protein kinase C is activated by this excessive diacyl glycerol, it's going to result in problems from multiple dimensions again, because protein kinase C is the one that's going to result in insulin resistance. If you remember the previous slide, I'd have told that diacyl glycerol causes insulin resistance. How diacyl glycerol causes insulin resistance? That's by activating the protein kinase C. And at the same time, protein kinase C can also increase the activity of a very important enzyme called NADPH oxidase. Once NADPH oxidase is activated, they are going to result in more reactive oxygen species and free radical production within the cell. Already the cell is having less NADPH, difficulty in coping up with the free radical stress. Coupled with that, this excessive diacyl glycerol and protein kinase C is actually activating NADPH oxidase, producing more free radicals, resulting in more and more damage in the cell. And what are the effects of excessive free radicals? Let us see in some time. And apart from that, this protein kinase C can also increase the production of VEGF, which is fairly important because that's going to increase the angiogenesis in the certain organs. And this increased angiogenesis is very, very important for development of certain important microvascular complications like diabetic retinopathy. And they're also going to increase the activity of TGF-beta, that is transforming growth factor beta. And this is very important for extracellular matrix deposition. And that in turn is important for development of one of the very important microvascular complications in diabetes, that is diabetic nephropathy. And apart from that, there can be macrovascular complications in diabetes that is due to increase in the production of PAI1, that is plasminogen activator inhibitor 1, which is a very important compound in the setting of development of procoagulant tendency and thrombosis. And they can also reduce the endothelial nitric oxide synthase enzyme, reducing nitric oxide production, and they can increase the endothelin 1 production, resulting in balance towards vasoconstriction and thrombosis once again. So these are the reasons for potential microvascular as well as macrovascular complications that are happening in diabetes mellitus, but there is a lot of evidence for development of microvascular than a macrovascular complication in this regards. Apart from that, you're not only going to produce excessive glyceraldehyde 3-phosphate, you are also going to result in production of excessive acetyl-CoA as well. Obviously, as a part of accelerated glycolysis, there are going to be production of accelerated acetyl-CoA as well. And this excessive acetyl-CoA will enter the TCA cycle where they are going to generate excessive energy-rich electron carriers like NADH and FADH2. Of course, what is the role of this NADH and FADH2? These contain high energy electrons which will be transported across the electron transport chain, which will be utilized to produce a proton gradient that will be used to synthesize ATP through an enzyme called ATP synthase. We know that already. But when you produce too much of this energy-rich compounds inside the cell, all this cannot be absorbed by the electron transport chain, resulting in spillover effect once again. And this spilling over effect, of course, is going to result in more generation of free electrons within the cell, resulting in more production of free radicals and reactive oxygen species in the cell. And there are going to be multiple effects of this reactive oxygen species and free radicals, which are talking about for a long time. But the first effect is denaturation of this glyceraldehyde 3-phosphate dehydrogenase enzyme. And this is quite sensitive to the free radicals. And of course, once this enzyme is inhibited or damaged, you're going to result in more glyceraldehyde 3-phosphate, producing more methyl glyoxal and more advanced glycated end products. And at the same time, you're going to result in more glycerol 3-phosphate, more diacylglycerol, increased activation of protein kinase C, and all these problems which we have seen already. And in short term, you can view all this process as physiological homeostatic mechanisms. Remember, only in chronic conditions of hyperglycemia are going to result in problems. But apart from that, in short term areas, you can view this as physiological homeostatic mechanisms. How? Because whenever there is excessive energy in the cell, you don't want any more glycolysis to happen. So that's why these free radicals are actually inhibiting the glyceraldehyde 3-phosphate dehydrogenase, where you need not produce more glycolysis, and you can shunt to energy storage mechanisms by producing more glycerol 3-phosphate to triacylglycerol, or you can result in stoppage of the glycolysis altogether. So you don't want energy anymore. And even the same thing can be viewed in the setting of this hexosamine pathway as well. Because you have too much of glucose inside the cell, more fructose 6-phosphate is produced. Because of that, the more glycosylation will happen and there will be reduced GLUT4 activity. And that can be viewed in short-term basis as a physiological homeostatic step, because whenever the cell is having more glucose, it don't want any more glucose. That's why indirectly your GLUT4 activity is reduced by more glycosylation, so that your uptake of glucose by the cell will reduce. So in a short term, all these mechanisms can be viewed as a physiological homeostasis, but in the long run, it's going to create a lot of issues by altering a lot of biochemical reactions within the cell. So there are some other effects of this free radicals and reactive oxygen species as well. For example, they can result in inhibition of certain other enzymes like endothelial nitric oxide synthase and prostaglandin I2 synthase, which is nothing but prostacyclin synthase, because PGI2 is nothing but prostacyclin. Both these are powerful. Nitric oxide as well as prostacyclin are vasodilators, anti-aggregating agents, which means antiplatelet aggregating agents, and they are very powerful protective agents against atherogenesis and thrombosis. Once these levels are decreased, if the nitric oxide levels are decreased and prostacyclin levels are decreased, they are going to favor atherogenesis and thrombus formation, resulting in macrovascular complications in diabetes. And at the same time, they can have multiple effects, other effects as well. Like for example, these free radicals are a very powerful inhibitors of this PDX1 mRNA. So there will be reduced production of this PDX1 protein. If you remember in the previous video, I was told PDX1 is one of the important transcriptional regulators for insulin gene. So which means for insulin production, you need this PDX1. If you have less PDX1, you will result in reduced insulin production from the beta cells. And this is a form of glucotoxicity on the beta cells itself, which is very relevant in the setting of diabetes mellitus type 1 especially, where excessive glucose for a long time itself can suppress the insulin production from the beta cells. And third mechanism is death of the beta cells. How they are going to produce death of the beta cells, even though you know that free radicals are going to cause damage on the cells, causing death, but there are some molecular mechanisms. Like for example, they can activate an enzyme called phospholipase C. We are talking about certain enzymes which are inactivated, but in this case, we are talking about enzymes that are activated. They can activate this phospholipase C, which is going to produce more inositol triphosphate and diacylglycerol. of course, this diacylglycerol will produce more activation of protein kinase C epsilon. That's going to result in more insulin resistance, more microvascular problems due to that high VEGF, which we have talked already enough. And this excessive inositol triphosphate once again can result in more calcium release into the cell from the endoplasmic reticulum. And this excessive calcium in the cell, in the cytosol, means it will increase the permeability of mitochondria to cytochrome C. You know where we are moving towards, apoptotic pathways. They are going to increase the permeability of mitochondria to cytochrome C. There will be leakage of cytochrome C in the cytosol. So increased cytochrome C in the cytosol will activate the apoptotic pathway by activating the caspase cascade. So once the caspase cascade is activated, you are going to result in death of the beta cell, resulting in apoptosis, and there will be reduction in the beta cell mass over a period of time, which means the hyperglycemia itself can cause death of the beta cells. That's why type 2 diabetes mellitus patients also will have reduced number of beta cells over a period of time. That is because of chronic hyperglycemia. There is a dictum that at the time of diagnosis of type 2 diabetes mellitus itself, there will be loss of beta cells by a ratio of around 30 to 50% approximately. The beta cells would have died already. That is because of chronic hyperglycemia in those individuals. And these two mechanisms which I have told you are going to contribute to the glucotoxicity on the beta cells itself, resulting in reduced insulin production by the beta cells because of the excessive glucose that is happening chronically. And apart from that, there will be fourth issue that is going to happen because of excessive free radicals, that is redox imbalance. What do you mean by redox imbalance? It's quite simple. Redox means it's reduction, oxidation, and it's always should be a balance. Now, free radicals are high, reactive oxygen species are high, which means oxidation is on the upper hand and reduction is lower. And coupled with low NADPH, already I told you that is consumed by the sorbitol pathway, there will be low reduction capacity in the cell. So there will be oxidative stress. That's what we call redox imbalance, resulting in more problems like increased calcium release from the endoplasmic reticulum and so on. We're going to result in more beta cell death and more problems in insulin production and release. So in short, as a summary, I can tell the entire problem in one single word. Because of hyperglycemia, you're going to result in energy stress. Too much energy that is produced inside the cell is also a problem. And that is the reason why you're getting so much of issues in chronic hyperglycemia. And there's another entity that you have to know apart from this chronic hyperglycemia and glucotoxicity, that is called lipotoxicity. I'm not going to discuss in a great depth and detail, but still it's important to understand. So diabetic patients are going to have high levels of circulating free fatty acids. Why they should have? Let's see. First of all, you know the circulating free fatty acids come from triglycerides, which are stored in the adipose tissue. And of course, free fatty acids to be produced, you need an important enzyme called hormone-sensitive lipase, which is actually under a strong inhibitory control by insulin. And what's going to happen in diabetes mellitus? Of course, in type 1 diabetes mellitus, there will be insulin deficiency. Because of that, you do not have insulin, so that your hormone-sensitive lipase activity will be very less, resulting in more production of free fatty acids. In type 2 diabetes mellitus, there will be insulin resistance. Because of that, you will have action of insulin that is defective on hormone-sensitive lipase, resulting in more activity of hormone-sensitive lipase, resulting in more generation of free fatty acids once again. Of course, diabetes mellitus is a condition where in the blood you will have more free fatty acids, irrespective of the type of diabetes mellitus. And this excessive free fatty acids will enter beta oxidation, because I told you in the beta oxidation of fatty acids, the most important regulated step is actually the lipolysis step by hormone-sensitive lipase. If that is not regulated properly in the setting of insulin resistance or insulin deficiency, you're going to result in more production of free fatty acids, and which also means that the beta oxidation will happen unregulated, and that will result in the production of more acetyl-CoA, which means you're going to result in more entry of acetyl-CoA into TCA cycle, more production of this electron-rich or high-energy electron carriers like NADH and FADH2, resulting in more production of free radicals and reactive oxygen species, and the problem is going to come back again in a much bigger way. So there are two things which you have to know. One is glucotoxicity, and second is lipotoxicity as well, which are happening in diabetes mellitus hand-to-hand. And the discussion will not be complete unless and until I talk something about this advanced glyceride end products and how they are formed first of all, we have to know that there is a very important highly reactive intermediate product called as carbonyl glyoxal. And carbonyl glyoxal is the one that is going to result in the development of products called as AG, that is advanced glycated end products. And what are the sources of this carbonyl glyoxal? Which means what are the things that produce this carbonyl stress inside the cell to produce this advanced glycated end products? One, we know already, it is the glyceraldehyde 3-phosphate. If it is in excess, they are going to result in production of more methyl glyoxal, which is a form of carbonyl glyoxal only. And second, you can have this carbonyl glyoxal whenever there is more lipid peroxidation in the cell. So lipid peroxidation is a way to produce this carbonyl glyoxal. Why? Because in diabetes you will have excessive free fatty acids. Along with that you will have more free radicals as well. Because you have more free fatty acids and more free radicals, they can cause lipid peroxidation. So which is a combination of both. And polyol pathway also to an extent can contribute greatly to the production of this carbonyl glyoxal intermediates, which are going to result in AG production. But more than that, what is important for your understanding is the fact that there is a principle called autooxidation of glucose, where glucose in excessive amounts can autooxidate itself, resulting in production of this carbonyl glyoxal and subsequent AG production. So what do you mean by autooxidation of glucose? It is, you need a protein, that's all. So in the reaction, you will produce a Schiff base initially, then there will be an Amadori rearrangement. Finally, you will result in production of this advanced glycated end product. So let us see how. And this formation of Schiff base is very important in chemistry in the 11th and 12th standard chemistry as well. But anyways, let us see in a much simpler way. And please understand the fact this is a non-enzymatic reaction. That's why it's called glycation. First, you're going to have glucose, which I can write in short as R, C, H and O, because glucose is going to have this aldehyde group because it's an aldohexose. And a lot of reducing sugars can undergo this Schiff-base reaction. What are reducing sugars? Your monosaccharides like glucose, fructose, then galactose, all are reducing sugars. And your disaccharides like maltose, then lactose are reducing sugars, but sucrose is not a reducing sugar. There are a lot of reasons for that, which we'll be seeing subsequently if you have time. But anyways, in this example, you're talking about glucose, glucose and glucose because you're talking about diabetes mellitus. And this is going to combine with the protein. I can write as NH2, which contains amino acid group, which is going to be combining with the R group, that is amino acid, which is a very integral part of a protein. And this reaction is something that happens non-enzymatically. You can see there is no enzyme that is involved in this reaction, which will result in removal of this H2 and O, nucleophilic attack. So H2O will be gone. So the resultant product is what we refer to as a highly active, unstable intermediate called as a Schiff base. So I can write as R, C, H and N and R. So this is what we refer to as a Schiff base, and this is a highly reactive and unstable intermediate. And this Schiff base in the setting of chronic and long-standing high levels of glucose can happen in hours to days. And over time, it will spontaneously rearrange. Because this is unstable, this has to rearrange into a new product called as Amadori product by a principle called as Amadori rearrangement. And Amadori rearrangement and Amadori product formation happens over a period of days to weeks. It takes some time to rearrange. And once this Amadori product is formed, you will result in the development of AG, which takes place over a period of weeks to months. Advanced glycated end products. Clear? And this whole process in forming the Schiff base to Amadori product, then to advanced glycated end products is what we refer to as a Maillard reaction, which can take place in many food compounds also, but this can happen in the body itself, resulting in certain irreversible damages to the certain proteins called as Maillard reaction. And anyways, no matter what, you have produced the advanced glycated end product at this step. So what is going to be the effects of this advanced glycated end products? That's what is the most important here. So this AG is going to work on the AG receptor, and this AG receptor is what we refer to as RAGE receptors. I think I have given a hint about these RAGE receptors in the hematology section itself. That's called receptor for advanced glycated end products. So once this AG binds with the RAG, you're going to result in subsequent downstream signaling pathways, which is going to cause a lot of troubles, like they're going to increase the TGF-beta production from the cell, which in turn is going to result in increased extracellular matrix deposition, and that's important for thickening of the base membrane, resulting in diabetic nephropathy. I told you already. And they are going to result in increased production of VEGF, which again is going to produce increased angiogenesis, which is in turn is going to be very important for the development of diabetic retinopathy, which we know that. And they're going to result in increased production of reactive oxygen species, increased production of PA1, both of which are very important for developing macrovascular complications like thrombosis, especially like CV and myocardial infarction, atherogenesis. And they can also produce increased nuclear factor for kappa B, which is a very important pro-inflammatory compound, and they can increase the production of RAGE receptors itself, which means binding of the AG with RAGE will result in increased production of RAGE itself, which will result in more binding with advanced glycated end products. So it's like a vicious cycle which is going to go on, and all these are the ones that are responsible for the development of the microvascular and macrovascular complications that you're seeing in diabetes mellitus. And at the same time, this advanced glycated end products itself, so let us put in green, the advanced glycated end products itself has some certain effects. For example, they can directly cross-link extracellular matrix proteins. They can directly cross-link the extracellular matrix proteins. For example, proteins like laminin, collagen can be directly cross-linked together by this AG, resulting in certain structural changes in these proteins. So which means they will be resistant to degradation. These cross-links will make them resistant to degradation. If they are resistant to degradation, there will be more extracellular matrix proteins which cannot be degraded. And that is also one of the main reasons why you get the thickening of the base membrane, which is very important in the setting of diabetic nephropathy. And this modified and cross-linked extracellular matrix proteins can trap certain non-glycated proteins. So they can also trap certain non-glycated proteins. What is more important for our clinical setting is that they can trap LDL particles, low density lipoproteins, which is very, very important for development of atherogenesis. That is also one of the reasons. This is not the only reason, but this is one of the reasons why at the same level of LDL, diabetic patients are going to have more problems compared to non-diabetic patients, because they trap more LDL, the LDL composition is a little different in diabetic patients. So all these things together will tell you that for the same level of LDL, diabetic patients are going to have more atherogenic risk compared to the non-diabetic patients. So you have so much of issues. That's why understanding about this advanced glycated end products is really, really important. And one of the important evidences for glycation that is happening in the body can be tested by using something called a HbA1c test, that is glycated hemoglobin. Remember, many books will call this as a glycosylated hemoglobin, but that's a wrong name. You should not call it as a glycosylated hemoglobin. It is a glycated hemoglobin, or you can simply call it as a glycohemoglobin. That's also okay, but the exact correct terminology is a glycated hemoglobin, not a glycosylated hemoglobin, because glycation is a non-enzymatic addition of glucose, and that is what is going to cause the AG. And a sample of how much glycation that is going on in the blood, which can actually predict the microvascular complications, macrovascular complications, is what we are going to test by using this HbA1c. That is glycated hemoglobin. So with this basic understanding, let us see what is the diagnostic criteria for diabetes mellitus. And what's the basis of giving such diagnostic criteria for diagnosing a disease like diabetes mellitus? It's not like an X, Y, or Z person giving a particular value telling that beyond which you're going to diagnose diabetes mellitus. There can be two ways to give a cutoff for diagnosing a disease. One is taking a mean and plotting it in the normal curve, and then finding out the standard deviations and telling the extremes can be taken as cutoffs for diagnosing a disease. Or second way where you can do is by taking a particular value and telling that beyond this value, you are going to have increased incidence of health-related problems. For saying diabetes, you're going to have cardiovascular problems or nephropathy or retinopathy, whatever it is. So that's how you give a value. It's not like some random person is going to give some random value telling beyond which you're going to have a diabetes mellitus. And at the same time, these values are changeable in the first place because a sugar of 110 would have been normal if the lifestyle is good. But if the lifestyle is bad, sugar of even 100 could be abnormal. So it all depends on the lifestyle and how the population evolves over a period of time, how our ecological system evolves over a period of time. So these values are highly changeable. So you have to understand that. So the same value may not be the case in 2035 probably, where the lifestyle may be even more bad, where the cutoffs for diagnosis of diabetes may come to 110 or 115 from the current cutoff of 126. We don't know. Or if the lifestyle is improving, even the cutoffs might increase to 135 as well. So we don't know what's going to happen in the future. So let us go and analyze what are the things that you are going to utilize to diagnose a diabetes mellitus. One is the glucose, of course, because high glucose, glucotoxicity, a lot of pathophysiological mechanisms we have described already. Second is the HbA1c. It's a very important evidence of how much glycation that is happening in the body, because HbA1c is a sample of glycation that is ongoing in the body. So this glycation causing this AGs is one of the very important reasons for micro and macrovascular complications in diabetes. So HbA1c will also tell you the risk of developing this micro and macrovascular complications apart from helping you in the diagnosis of diabetes mellitus. In 2010, ADA as well as WHO endorsed the utilization of HbA1c in the diagnostic criteria for diabetes mellitus. So this was the criteria that was given in 2010 and it was not revised after that. So let us see what is the diagnostic criteria. So first we are going to use the HbA1c, which is the new addition in 2010. Before that it was not there. And HbA1c's advantage is it can be done at any time. No problem. It can be done randomly because it's going to tell only the mean blood glucose. It has poor correlation with both fasting as well as postprandial. Rather, it will tell you a mean glucose. And it's going to tell the mean glucose over the period of last 2 to 3 months, which is nothing but the lifespan of the RBCs. And that's also one reason why HbA1c can be affected by the lifespan of the RBCs. And we are going to have fasting blood glucose, which means you should be in at least 8 hours of fasting before giving the sample. And we have a standard 2-hour 75 gram oral glucose tolerance test, where you will be asked to take 75 gram of glucose solution, followed by 2 hours later you will be asked to come back and again you have to give a blood test. And remember, whatever the values I'm going to tell will be applicable only for venous blood glucose or plasma glucose, not for the capillary blood glucose. Capillary blood glucose values are little different compared to the venous glucose, and all these standards are set for the venous glucose only. That's the plasma glucose or the blood glucose. So let us go to the values. FBG values less than 100, 2R values less than 140, HbA1c values less than 5.7%. Interpretation is likely normal. You are not suffering from any problem. So if the fasting levels are in the range of 100 to 125, 2R OGTT values are in the range of 140 to 199, and HbA1c values in the range of 5.7 to 6.4%, interpretation likely that the patient is suffering from pre-diabetes, which means these patients are at high risk of developing diabetes mellitus. And of course, if it's fasting that is abnormal, that is called as impaired fasting glucose. And of course, if the 2R OGTT is abnormal, that's referred to as impaired glucose tolerance. If both are abnormal, we'll be calling as impaired fasting glucose and impaired glucose tolerance. But nevertheless, the final statement that you're going to mention in your case sheet is pre-diabetes. Or if the fasting levels are more than 126 and the postprandial values, that is 2R OGTT values, are more than or equal to 200, or if the HbA1c values are more than or equal to 6.5%, we are going to diagnose that patient as diabetes mellitus. But very importantly, you have to understand just one value, if it's abnormal, that's enough to diagnose diabetes mellitus. It's not like you should have all the 3 values to be abnormal. But there's one small thing in that. So if there is only one value that is abnormal, I'm not telling you cannot diagnose diabetes. Still, you can diagnose diabetes mellitus, but you should prove at least 2 times. If both the values are abnormal, that fasting is also abnormal and postprandial is also abnormal. So in that setting, you can straight away diagnose diabetes mellitus with no need for the repeat sample. Clear? And apart from that, WHO gave another criteria in 2011 where they told random blood glucose of more than or equal to 200 along with classic symptoms of diabetes mellitus like polyuria, polydipsia and polyphagia can be used to diagnose diabetes mellitus. This is very important in the setting of poor resources like India, where you might be able to miss the patient if you ask the patient to come for the next day, where they may not come and turn back. So where you have to make the diagnosis very fast. And this is very, very true in the setting of type 1 diabetes especially, because the patient is coming with some symptoms like polyuria, polydipsia and polyphagia, and you are seeing a random blood sugar of more than 200, you have to immediately make the diagnosis of type 1 diabetes mellitus as far as children are concerned, because any delay in the diagnosis and the patient is presenting with symptoms means even within 6 or 8 hours, if you delay the insulin treatment, the patient might go for diabetic ketoacidosis and they might come in a much bad position. So that's the reason why the diagnosis should be fast in these cases, especially in the setting of type 1 diabetes mellitus. That's why WHO gave this additional criteria. This is the 4th criteria. So how can I diagnose diabetes mellitus? Fasting blood sugar of more than or equal to 126, 2-hour OGTT values of more than or equal to 200, HbA1c values of more than or equal to 6.5%, or random blood glucose value of more than 200 with classic symptoms of diabetes mellitus like polyuria, polyphagia or polydipsia, and or anyone if you have also you can diagnose diabetes mellitus according to WHO. After knowing the diagnostic criteria for diabetes mellitus, it's also prudent to understand HbA1c in a little bit more depth, because every guideline in the world is going to endorse the utility of HbA1c currently, because HbA1c is something that is going to correlate highly with your risk of micro and macrovascular complications. At the same time, the current glycemic target standard is HbA1c. Even though the targets are different currently compared to the previous strict target of 7%, now we are having a flexible target for a particular patient and it should be patient-centered. But nevertheless, still HbA1c is the standard glycemic target for the control of diabetes. And at the same time, HbA1c is the standard monitoring tool for diabetes mellitus. And at the same time, this is the one that's going to tell you the risk of micro and macrovascular complications in the future, which means this is the one that best correlates with the development of micro and macrovascular complications. So many things are there about HbA1c. That's why it's important to know about HbA1c. First of all, what do you mean by HbA1c? It is a glycated hemoglobin where glucose freely diffuses into the RBC and attaches with the hemoglobin by a non-enzymatic process. That's why we call it as a glycated hemoglobin, or some textbooks give a glycohemoglobin, but avoid the term glycosylated hemoglobin as I told you before. And we know the normal value less than 5% is regarded as normal. Values in the range of 5.7 to 6.4% is considered to be pre-diabetes, and values more than or equal to 6.5% will be deemed as diabetes mellitus. All right. And what HbA1c is going to tell you, basically, it's going to tell you the mean blood glucose, which means the average blood glucose over the period of last 2 to 3 months. So why we are telling it's going to tell the average blood glucose over the period of last 2 to 3 months? Because this will indicate the involvement of the RBC lifespan in this. So that is why when you're talking about a falsely elevated or a falsely low HbA1c than predicted, you think about conditions that alter the RBC lifespan, because when you alter the RBC lifespan, HbA1c levels also may be altered, because the hemoglobin's lifespan in the RBC will be dependent on the RBC lifespan only. So that's the reason. So how can you tell the mean blood glucose for a particular value of HbA1c? The formula is very simple. It is 33.3 multiplied by a particular HbA1c value minus 86 milligrams per deciliter. So for a given HbA1c, use this formula, you will be getting the mean blood glucose over a period of 2 to 3 months. But many students, including practitioners, do ask one question. Mean blood glucose means whether you are talking more about a fasting blood glucose or a postprandial blood glucose. Remember the fact that we are talking about a mean blood glucose over a 24-hour period for the last 2 to 3 months. We are not talking about a fasting or a postprandial glucose. But because many people are persistent with that question, which I feel it's illogical, I will tell an illogical answer that it's going to correlate with the fasting blood glucose more than the postprandial blood glucose. And this is something that was given in earlier studies in 2006 and 2007, but that's something illogical to be precise. But we are going to find out a mean blood glucose rather than its correlation with the fasting blood glucose or the postprandial blood glucose. Because there was some studies in the early stages which told that fasting control is associated with the better control of HbA1c also. Even if postprandial is fluctuating a little bit, it is not going to affect your HbA1c that much, and that really makes sense also, because fasting time is the one that's going to persist for a longer period of time. Your postprandial time is only a fluctuating time. It's only going to produce a small spike in your blood sugars. But the baseline, that's the fasting level, is the one that's going to persist for a long time during the day. And that's why the mean blood glucose is also a little and mostly dependent on the fasting blood glucose rather than the postprandial excursions of blood glucose levels. So, but in general, it's an illogical question, but I can tell it more correlates with the fasting blood glucose than the postprandial blood glucose. And as I told you, it's heavily affected by the lifespan of the RBC, so you have to be careful in assessing the values. And one important advantage is the fact that it can be monitored just once in 3 months. So you need not monitor less than 3 months, before 3 months, because that doesn't make any sense again, because for new RBCs to come and for them to get glycated and to reflect the actual value, it takes around 3 months. So it's better to test once in 3 months, that's more than enough. And more importantly, in exam, where they will concentrate is the fact that, what are the areas where you have more HbA1c than you're going to expect, and what are the areas where you're going to have less HbA1c than you're expecting. So we're talking about false values, more HbA1c than expected and less HbA1c than expected. So let me tell you a condition where you're going to have more HbA1c than expected. You're having a normal blood glucose, which means fasting sugar is normal, postprandial sugar is normal, but you're having a very high HbA1c levels. What is the probable reason? Where you'll expect, in the sense, what is the meaning of less HbA1c than expected? You are having a high blood glucose, like fasting of 200 or 250, postprandial of 300, 350, but you're having HbA1c of only 5.9% or 6%, which means you're having actually a lesser HbA1c than you're actually expecting in that particular patient. So where you're going to get that? So first scenario is based on the RBC turnover and RBC lifespan. There are some conditions where RBC turnover will be very less. For example, post-splenectomy. Obviously, in this condition, the RBC lifespan also will be a little high. So which means the same RBC is going to be circulating for a longer period of time in the body. So that's why the the glycated hemoglobin also persists for a longer period of time, leading to a cumulative accumulation of glycated hemoglobin, giving a relatively higher value. So you have to think about that. And apart from that, the same thing happens in iron deficiency anemia, megaloblastic anemia, vitamin B12 and folic acid deficiency, where you are having a low RBC turnover. So the same RBC is going to circulate for a longer period of time. So that is the reason why you can have a cumulative accumulation of glycated hemoglobin in the RBCs, resulting in a more than expected value of HbA1c. And apart from that, you can see in other conditions like anemia of chronic disease and anemia of chronic kidney disease. Both are low turnover conditions as well. And aplastic anemia. So again, this is a low turnover condition. And there are certain hemoglobins which will result in low RBC turnover, like HbF, the destruction will be very less. And in HbH, once again, it's a low turnover condition. Similarly, where you are going to expect less HbA1c than expected, when you are going to have a more RBC turnover. In the sense, lifespan of the RBC is considerably less in this scenario. The first thing that you have to tell is hemolytic anemias. Anything that produces hemolysis, like for example, mismatched transfusions, G6PD deficiencies, hereditary spherocytosis, sickle cell disease, anything that produces hemolysis is going to result in more RBC turnover, so that you will result in lesser HbA1c than you are going to expect. Because the RBCs that are already glycated will be destroyed very fast, and new and fresh RBCs will be coming from the bone marrow, which takes time to get glycated. And if you take the blood sample, those new RBCs will not have adequate glycated hemoglobin, resulting in a falsely lower values. So this can happen in the setting of hemorrhage. So post-hemorrhage, there will be a reticulocyte response, which is a fresh RBC, more turnover. So similar effect. Pregnancy, of course, you will have a more RBC turnover. Then high altitude, of course, because of hypoxic erythropoietin response, you are going to have a more RBC turnover. So where you have to be careful when you are practicing in hilly areas. And during treatment, especially when you are treating an iron deficiency anemia with iron, there will be a reticulocyte response and increased turnover. And in megaloblastic anemia, when you are treating with B12 and folic acid, once again you have to be careful where treatment will increase the RBC turnover. Then when you are treating an anemia of chronic kidney disease or anemia of chronic disease with erythropoietin therapy, which is going to remove the block and increase the RBC turnover. So in these settings, you might have a lower HbA1c than expected. And at the same time, certain dyshemoglobins like HbS and abnormal hemoglobins like HbC and HbD, they might be having a higher RBC turnover, resulting in falsely lesser HbA1c than you're going to expect. And there are certain conditions that can produce assay interference. Because of that, you might have a false value of HbA1c. In the setting of more HbA1c than expected, you can think about hyperbilirubinemia, especially in the range of maybe 30 or 35 bilirubin. Hyperbilirubinemia can interfere with the assays, and hypertriglyceridemia also can interfere with the assays. And it has to be very, very high, like triglycerides of 2000, 3000, bilirubin of 30, 25. So they can interfere with the assays, and they can produce falsely elevated HbA1c values than expected. And in the setting of less HbA1c than expected, falsely low values, you can think about consumption of massive dose of vitamin C or vitamin E, especially when it's taken in very high doses, that can interfere with the assays, resulting in poor detection of HbA1c, causing lesser HbA1c, that is falsely less HbA1c than expected. Apart from that, there are patient-related factors that can produce these issues. What are the patient-related factors? Like the most common reason, in fact, where you see a more HbA1c than expected is patient trying to falsify the reports. So what do you really mean by patient trying to falsify the reports? That the patient is going to go to the lab and tell that give me a higher HbA1c, not like that. So here in this condition, this is a common scenario, especially in many countries where patients come once in 3 months or 6 months. They would have been maintaining a very bad lifestyle for the last 3 to 6 months, taking a very bad diet, not exercising. And they know that their sugars are going to be high, and they know that they're going to meet the doctor in the next maybe 2 to 3 days. So that appointment is due. And before coming to the doctor, they go on diet. They don't eat anything. They just don't take any food with high glycemic index, and they control the sugars, but that control will be reflecting only in the fasting and postprandial. So they'll have an absolutely normal or a slightly elevated fasting and postprandial, but if you see the HbA1c, it will be sky high, it will be 11-12%. So you can clearly think about patient falsifying the reports. You have to ask that how long you have been in control. Clearly, they will tell only 2 to 3 days in the last week. Apart from that, they will not have been controlled in the last 3 to 6 months, where you can easily catch them by using the HbA1c values.